Good morning, everyone. My name is Newton Castro, and I welcome you to the third session of the third workshop on inflammation. Our topic today is neurodegenerative diseases, and of course, it's all about neuroinflammation. I have the great honor to introduce to you the first speaker, and this morning, Professor Francisco Javier Quintana, who is professor of neurology at Harvard University, associated also with Brigham and Women's Hospital and the Broad Institute. Professor Quintana is a highly cited scientist. He's amassed over 14,000 citations and is continuously having over 1,000 citations a year, at least in the last decade. So this is really still going up. And of course, this is because of his major contributions in the area of neuroinflammation, specifically in revealing mechanisms of central autoimmune neuroinflammation. So the major contributions are in the role of environmental factors in neuroinflammation, like aryl hydrocarbons, and more recently, the microbiota, of course. And much of this beautiful work has been using the mouse model of multiple sclerosis, the autoimmune, experimental autoimmune encephalomyelitis. It's interesting that the most cited paper by Dr. Quintana is shared also with our third speaker, Dr. 𝗨𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 is a nature paper in 2008 that has beautiful contributions to, specifically in the area of the aryl hydrocarbons in neuroinflammation. So I leave the room to Professor Quintana, and please welcome, and I hope you enjoyed the talk.
Thanks, Newton, for that very nice introduction. And actually, thanks to the organizers for giving me the opportunity to present our work here. It's actually a joy to be able to present among friends and to be among friends, at least virtually. So what I'm going to do today is I'm going to talk about the role of astrocytes in CNS inflammation. And in particular, our work relates to one disease, or has been mostly focused on one disease, multiple sclerosis, and its disease and its preclinical model experimental autoimmune encephalomyelitis, or EIE. And this is a work, and this is a model with which I'm sure you're very familiar. You have people doing very good work there, such as Alex Basso, who's going to be talking in a few minutes. So when we think about MS and when we think about its model, EIE, we usually think about T cells being induced in the periphery, right? T cells reactive against myelin and other brain proteins. They are then reactivated in the CNS, and once they get to the brain and the spinal cord, they cause damage to axons and myelin. And this, from a clinical point of view, is a process we understand quite well. And there are multiple therapies that can actually interfere with these multiple steps of MS pathogenesis. However, when we think about MS, I think those T cells and those peripheral cells are going to do in the CNS is they are going to activate CNS resident cells, cells that are actually intrinsic to the brain and the spinal cord, such as microglia and astrocytes. And that's important because once those cells get activated, additional pathogenic factors, additional pathogenic mechanisms are triggered that are going to lead to neurodegeneration, are going to lead to CNS inflammation, and with two important corollaries. First of all, we do not have therapies available in order to control the pathogenic activity of astrocytes and microglia. And also, the pathogenic activities of astrocytes and microglia in multiple sclerosis are likely to be shared and are likely to contribute to the pathology of other neurologic disorders such as Alzheimer's disease, Parkinson's, so on and so forth. Because of that, it's very important to understand what are the pathways that regulate astrocytes and microglia. And our goal has been, or our lab has been mostly focused on astrocytes. Astrocytes got their name because they look like stars. They are star-shaped cells. And indeed, when we think about them, they were known to do, and they do perform multiple functions in health. they can actually participate in development and the regulation of the blood-brain barrier, of synapses, and also under homeostatic conditions, astrocytes are going to provide metabolites and neurotrophic factors that kept the health of neurons and allow neurons to function properly. Many of these homeostatic functions are impaired in the context of CNS pathology, are impaired in the context of MS. On top of that, astrocytes in the context of pathologic conditions gain new functions, gain pathologic activities associated with the recruitment of inflammatory cells from the periphery into the CNS, the induction of neurotoxic and pathologic activities in CNS resident cells such as microglia, and even astrocytes can show intrinsic neurotoxic activities. So astrocytes, on the one hand, in the context of pathology, are going to stop doing what they should be doing, and on top of that, they are going to gain additional pathologic activities. So when we think about these good and bad things that astrocytes do, it's important to keep in mind some very old findings made by Santiago Ramón y Cajal more than 100 years ago. The first finding I want to bring up is the fact that more than 100 years ago, he was able to identify multiple astrocyte subsets, at least in terms of the phenotype of these astrocytes under the microscope. So the first thing I want you to keep in mind is there's not one single homogeneous population of astrocytes, there are multiple subpopulations that we should consider. The second observation that Santiago Ramón y Cajal made was the fact that astrocytes are highly interconnected. If you look at these old drawings, you can see that astrocytes are connected with other astrocytes and also with other cells within the central nervous system. So then the question is, how are those connections, right? How is that cell-cell communication affecting specific astrocyte subsets and eventually affecting CNS physiology? So with that in mind, the first thing we've done is actually, I will briefly summarize what we've done in the past, and in particular, what we've done was actually to implement bulk and single cell technologies in order to identify novel subsets of astrocytes, both in MS clinical samples, basically in tissue from MS patients, but also in multiple clinical models, preclinical models of MS, such as the EIE model in B6 mouse or in the NOD mouse strain. So when we perform these studies, our goal is always to identify novel astrocyte subsets. Here you can see multiple populations defined based on the transcriptional signature. But then what we want to do is for each one of those populations to identify the molecular mechanisms that control them. And the reason for that is because those mechanisms can, on the one hand, become candidate targets for the modulation of this one disease, of these one astrocyte subsets in the context of disease. But the other important reason why it is central for us to identify which molecular pathway controls each one of these astrocyte subsets is that if we know each one of these pathways, we can perform in vivo perturbation studies, in vivo cell-specific perturbation studies. In particular, we use CRISPR for that. And that allows us to investigate and interrogate the function of these astrocytes. In other words, is this astrocyte subset associated with the improvement or the worsening of EIE? Does it drive disease pathogenesis? Does it suppress it? So using this approach, what we have done is identified at least 2 astrocyte subsets, at least 2 astrocyte populations that perform quite opposite roles in the context of inflammation. And the reason why I want to summarize those subsets to you is because they allow us to understand and to visualize how broad are astrocyte functions in health and disease, and how really heterogeneous can be each one of these astrocyte populations. The first population we identified is actually driven and controlled by a transcription factor called 𝗏G, which is a small member of the 𝗏 family of transcription factors. This is a transcription factor that controls the epigenetic status of this specific astrocyte subset, and by doing so, literally really controls which genes are expressed or not. And that has important implications because this astrocyte subset has a decreased ability to support the metabolic needs of neurons. Basically, it's less efficient in providing lactate to neurons in order to support their metabolism. In addition, this subset of astrocytes is characterized by the activation of XBP1 and other signaling molecules associated with the unfolded protein response. And that's important because many of the members of that signaling pathway, many of the members of the unfolded protein response have been identified as candidate targets for therapeutic intervention. We have small molecules that can modulate them. The last point I want to make about this astrocyte subset is that it actually seems to be driven by GM-CSF produced by a specific subset of T cells. So this is a clear example, and I want you to keep that little line of thought with you, that some T cells, right, or some cells recruited from the periphery can signal to astrocytes via the production of specific molecules such as GM-CSF in order to control the differentiation and drive their polarization into highly pathogenic subpopulations. This is one example I wanted to share with you. The second example I wanted to share with you is actually a population that seems to be doing exactly the opposite. And indeed, these two populations seem to be balancing inflammation within the CNS. The second population we identified, right, is characterized by the expression of the pro-apoptotic molecule trail in the surface. And to cut it short, what this astrocyte subset does is it literally induces T cell apoptosis as those T cells try to invade the CNS. So by doing, by inducing T cell apoptosis, this is an anti-inflammatory subset of astrocytes. The last point I want to make about this subpopulation of astrocytes is that it's induced in particular by NK cells that produce interferon gamma. So here you have another mechanism of cell-cell communication that seems to be inducing a very specific phenotype or subset in astrocytes. So if we step back for a second, right, I gave you an example of two of many astrocyte subsets that play quite opposite functions in the context of inflammation. As I mentioned, this is just two of many. There are probably other subsets of astrocytes in the CNS. And one point that seems to be a common theme in terms of the regulation of astrocyte phenotypes and subsets is the fact that these subsets seem to be controlled by cell-cell interactions. And just talked about how astrocytes are regulated by their interactions with T cells or NK cells. We have previously shown how astrocytes are regulated by microglia, or how astrocytes can crosstalk with neurons and oligodendrocytes. So that actually brings up the question. I mean, first of all, it highlights how important are cell interactions for the control of astrocyte function. But then actually that brings us the question, or brings up the challenge of how can we investigate cell-cell interactions in the CNS in a physiological and comprehensive manner? And this is very important because many of us have found that working with astrocytes in vitro is really prone to induce lots of artifacts. We cannot really investigate physiologic responses in vitro when we focus on astrocytes and microglia. But then the question is, how can we do it in vivo? So in order to do that, we developed a new technique that we call rabbit-seq, which is basically a method that uses the rabies virus as a way of inserting a genetic barcode in cells that are interacting in vivo. We can then isolate those cells, perform single-cell sequencing, detect that barcode, and use that barcode to identify cells that were interacting in vivo. So let me show you some examples of how that technology is working. We used this tech first in order to investigate cell-cell interactions in the CNS of 𝗖𝗓 mice. And in particular, we focus on the interactions that involve astrocytes because we wanted to focus in these initial studies on how other cells could control astrocyte phenotypes and what are the mechanisms that mediate those cell interactions. When we perform these studies, the first piece of data you get is single-cell RNA-seq data similar to the data that you get in other single-cell experiments. The only difference is that if you look at these t-SNP plots, right, we know that in these t-SNP plots, each one of the dots represent a different cell. When we do rabbit-seq, each one of these dots is actually annotated with a barcode that tells us which dots are interacting with which dots. In other words, we can turn a U map like this into an interaction map like this one. And these are actually interaction maps showing us the interactions of about 500,000 of cells within the CNS of 𝗖𝗓 or naive mice. And what you can see here is obviously the overall structure looks very different, but we can actually get into the details of each and every interacting cell. And when we do that, we can really obtain very valuable information. First of all, we can identify how cell-cell interactions, and we can quantify how cell-cell interactions are affected in the context of inflammation. In other words, we can tell that in the context of a naive mouse, right, astrocytes mostly interact with other astrocytes and microglia. However, the moment you induce 𝗖𝗓, additional interactions are established, not only with other cells of the CNS, but also with cells recruited to the CNS in the context of inflammation. Not only do we change in the context of inflammation the cells with which astrocytes are interacting, we also change the mechanisms used to mediate those interactions. Hence, when we perform rabbit-seq in the context of 𝗖𝗓, we can identify interacting cells, we can identify interaction mechanisms between those cells, and we can also go downstream and identify the effect that those interactions have on each cell type involved, the astrocyte and, for example, microglia. So we perform those studies, we perform those analyses, and in particular, we decided to focus on microglia and on macroglia astrocyte interactions. And in particular, we decided to focus on those interactions of astrocytes that showed the most pro-inflammatory disease-promoting phenotype. In order to do so, basically, we analyzed our single-cell RNA-seq data set. We identified within that data set those astrocytes that show the most pro-inflammatory transcriptional programs. And then we looked into our interaction data set and looked for the microglia connected with those astrocytes. Once we identified those microglia, we identified the molecular mechanisms mediating those astrocyte microglia interactions. And to our surprise, on the very top of our mechanisms of astrocyte microglia interactions, we detected a family of signaling pathways, which is called axon guidance pathways. These are actually pathways that are usually associated with development, but which are not really thought to play a significant contribution to CNS inflammation. So our data would suggest that pathways that are associated with CNS development are co-opted and reactivated by astrocytes and microglia in the context of inflammation. The second observation we made is that when we dig deeper into these axon guidance pathways, the one that was most activated was an axon guidance pathway involving the F receptor and its ligand ephrin. So to cut it short, the F family of receptors is actually a family of tyrosine kinase receptors. Here you have a tyrosine kinase domain, and that will become important afterwards. This is a family of receptors that is membrane bound, as you see here. And indeed, our studies identified FB3 receptor expressed in astrocytes as a mediator of astrocyte microglia interactions. The FB3 receptor interacts with a ligand, with a membrane-bound ligand, which is called ephrin B3. And what is interesting about this ligand is that ephrin B3 also has a signal transduction domain. In other words, when you have an interaction between astrocytes and microglia via FB3 and ephrin B3, both cells involved are likely to change their phenotypes, because in both cells, you're going to trigger intracellular signaling. In astrocytes, via the tyrosine kinase domain, and in microglia, via less characterized signaling pathways. So with that in mind, what we decided to do was to further investigate the role of FB3 expressed in astrocytes and ephrin B3 expressed in microglia as potential mechanisms regulating CNS pathology and EIE and MS. The first study we did was actually to just analyze CNS tissue from MS patients. And basically in those studies, we were able to detect a significant upregulation of FB3, the receptor, in astrocytes, and ephrin B3, the ligand, in microglia. Just note that this seems to be mostly detected in the lesions, and I'll get back to that later on. We then validated these findings in cell-specific gene inactivation or gene perturbation studies in EIE. So if we inactivate FB3 in astrocytes or its ligand in microglia, we get a significant amelioration of EIE. And that goes along with a significant decrease in the transcriptional programs of astrocytes associated with their pathogenic activities. We then actually went on and analyzed deeper what's going on here. And to cut a long story short, we found that FB3 signaling actually controls mTOR. And when mTOR is controlled, the activity of mitochondria is controlled. And in particular, FB3 signaling induces a mitochondrial reprogramming that results in mitochondrial dysregulation and an overproduction of mitochondrial reactive oxygen species that can trigger multiple pro-inflammatory signaling pathways. So these interactions between a microglia and astrocytes mediated via ephrin B3 and FB3 trigger signaling pathways that result in the activation of astrocyte pathogenic transcriptional programs via mitochondrial reprogramming. As I mentioned, when cells interact and when these cells interact via ephrin B3 interactions, microglia are also going to change the responses because there's a signal transaction domain here, and these are membrane-bound ligands. Indeed, in our studies, we detected a significant effect of ephrin B3 in boosting NF-kappa B activation and consequently in boosting transcriptional programs in microglia driven by NF-kappa B, which are known to promote CNS inflammation and neurodegeneration. The last point we wanted to address was whether we could target this pathway therapeutically. And for that, we exploited the fact that, as I mentioned, this intracellular domain of FB3 is actually characterized and involves a tyrosine kinase domain. So we screened a small molecule library, a library of small molecules which are actually CNS permeable. And our studies identified a small molecule, an inhibitor of this tyrosine kinase activity, which we named A38. If we administered A38 to mice, we get a significant suppression of EIE, either if we use this B6 model or if we use the NOD EIE model with which Basso worked a lot. And if we analyze the transcriptional program of astrocytes, we get a significant suppression of the pro-inflammatory responses of astrocytes when we treat mice with A38. We can also inhibit pro-inflammatory responses of human astrocytes in vitro, or even iPS-derived human astrocytes in vitro. But I don't have the time today to show this study. The final observation we made when we used this 𝗏𝗏𝗃 approach is that the second axon guidance pathway we identified as significant upregulated was one mediated by semaphorin. In particular, we found that semaphorin 4D expressing microglia can be activated or can activate plexin B1 and plexin B2 in astrocytes, and that also triggers multiple pro-inflammatory responses. And conversely, if we actually inactivate these interactions, there's a significant amelioration of EIE. One minor point I want to make is that these semaphorin 4D plexin B1-2 interactions seem to involve different subsets of astrocytes and those driven by FB3 signaling, because they seem to involve mostly astrocytes located not only in the lesions, but also astrocytes located in the normally appearing white matter of MS patients. And indeed, when we analyzed single cell data that we generated from MS patients, we see that different astrocyte subsets and microglial subsets are involved. So then I'm going to summarize what I showed you today. First of all, we developed this new technique, which we called 𝗏𝗏𝗃, which allows us to study CNS cell interactions in vivo. I showed you its application for the study of CNS interactions involved in astrocytes, but it can also be applied to study other cell interactions in the CNS, not only those involving astrocytes. And also, we have recently upgraded this technique. We call it now 𝗏𝗏𝗃 2.0, in order to be able to use it with wild-type mice, living non-human primates. We have run some very interesting studies in macaques, and also even with human tissue. And I think that's where it shows its most potential. For example, we have used it to study the tumor microenvironment of glioblastoma. And indeed, we did that using clinical samples from glioblastoma patients. So whatever we find is really the disease, is not a preclinical model. And we use that in order to identify the interactions between tumor cells with, for example, astrocytes, microglia, and other cells. These are interactions we recently summarized. That has led us to identify novel mechanisms by which the tumor tries to induce a very immunosuppressive tumor microenvironment in the brain. And we are actually performing in vivo validation studies in order to identify what we believe are going to be novel therapeutic targets. Second point I want to make is 𝗏𝗏𝗃 led us to identify novel astrocyte microglia interactions. And the point I want to make is we usually think about these two cells talking about soluble factors. The data I show you today identifies FB3 and FMB3 as actually membrane-bound factors, suggesting that these two cells can establish very tight interactions in vivo. And indeed, we were able to capture some of those interactions by electron microscopy. And now we're really trying to identify what other molecules might be mediating those interactions. And finally, just to summarize, specifically in the studies I showed today, these studies led us to identify two novel mechanisms mediating astrocyte microglia crosstalk, FMB3 and FB3, plexin B2, and semaphorin D4. And then in particular, we identify one small molecule that can interfere with those mechanisms and which we believe is going or might lead to novel therapeutic approaches for neurologic diseases. And with that, I would like to thank you again. It's really such a pleasure, such an honor to be able to meet you. Some of you have been friends for almost 20 years now. And I just want to finish by acknowledging those students, those scientists in the lab that actually ran these projects: Mike Wheeler, Cristina Gutierrez-Vázquez, Ian Clark, and Lily San Marco. And with that, I'd be happy to take any questions you might have.
Okay, thank you very much, Dr. Quintana. Beautiful talk. I already have a few questions that were made during the talk, and I'm happy to take. We have time for at least a couple of questions, I think. So Josiane Neves asked in StreamYard whether you mentioned that you could target individual astrocyte subsets therapeutically. So the question is, how can you do that for neurological diseases?
So the idea is, that's a great question. So what we are trying to do is basically, that's where we try to identify the molecular mechanisms that control these subsets, because then we can define or design drugs targeting each one of those specific subsets. FB3, and in particular the A38 inhibitor that I showed you, is an example of what we believe are going to be several subset-specific therapies. That's where we're going to.
Okay. And 𝗙𝗂𝗂𝗏 also made a couple of questions. I think they were directed to some specific point of the presentation. So when you say cell-cell interactions, did you mean direct contact?
Yeah, so the interactions we detect involve both direct contact, as the ones I showed today, but also soluble factors. We were able to recapitulate, for example, previous interactions that we described involving secreted VEGF or secreted IL-10, just to give you an example.
And he also asked whether you did consider microglia phagocyting virus or even dead infected cells in their role in these interactions. Yeah.
So that's something that we can actually, that we have actually been able to rule out. So that's actually something we controlled for.
Okay. Aline Ribeiro also asked if it's known at which point of the disease the therapeutic is successful. Perhaps she's referring to multiple sclerosis.
So we believe, so that's a very important point. In the context of MS, astrocytes and microglia are thought to drive disease pathogenesis for the progressive phases. So that's the stage of the disease we would like to target. Yet, their deregulated activities start at the very beginning of the disease, are triggered during inflammation. So our goal would be to start treating early as a way of arresting progression or even preventing progression.
Okay, I have a quick question too. All this new methodology that helps us address the transcriptome directly, and even the ones that address the interactions like your rabies technique, they seem to be taking pictures. And when we want to know is who drives who, so we need more real-time information. Are you working in this area too, like imaging brains in which the cells are live labeled, and to just be able to address that? Yeah,
so what we literally just completed is a mapping of literally the whole CNS by cytotranscriptomics. So with a method that gives us subcellular resolution. So literally the idea is to map this and even identify novel interactions and localize them to specific areas of the CNS with these approaches.
Okay, so I think we need to cut now because of the next speakers. I think I have one or two questions that I'll try to send to you later that I still.
Super.
So I apologize for the ones that couldn't answer right now. So thank you very much, Francisco. And now let's move on to our next speaker. It's my great pleasure to introduce 𝗙𝗂𝗂𝗏, who is a professor of pharmacology at the School of Medicine of the State University of São Paulo, Ribeirão Preto. And 𝗙𝗂𝗂𝗏, most of you probably have heard of, is one of our most prolific and relevant young scientists with enormous contributions in the area of neuroinflammation. The difference is now we are focusing more on peripheral nervous systems because 𝗙𝗂𝗏 is mostly interested in pain and mechanisms of both physiological nociception and hypernociception associated with inflammation. 𝗙𝗂𝗏 is extremely prolific. So to give you an idea, since 2020, he's co-authored more than 50 papers. This is just during the pandemics. Of course, many of his papers have amassed lots of citations, so there's relevant papers in this area. So he's contributed in proposing new and discovering new targets, so studying new cytokines and their receptors and mechanisms of interaction. So thank you very much, 𝗙𝗂𝗏, for coming. And this room is yours now.
So thank you very much, Newton, for the introduction, very kind introduction, and thank you also to the organizers, especially Claudia Benjamin, that invited me to give this talk. As Newton said, my lab is mainly focused on to understand the neurobiology of pain. And especially we are very focused on the neuroimmune glia interactions and how these interactions can contribute to pathological pain. And at the end of the day, the idea of the lab is to identify novel targets, as Francisco Quintana said, related to multiple sclerosis. In our context, we are trying to identify targets for the development of chronic pain, which is a very important medical need. So what we know is that the analgesics that we have in clinics are not so effective, and normally they have several side effects. So although we know our knowledge about the neurobiology of pain has grown in the last couple of years, we still need better analgesics. So first of all, I'd like to introduce a little bit about the field. So probably everybody knows very well about pain, which is a protective mechanism that we have. And but the problem in terms of pain is that usually, as I mentioned, pain is associated with injury, a peripheral injury or an organ injury. And normally, when we recover from this injury, we also have a reduction of pain. So this happens normally in our life. The problem is that some individuals or some persons that has trauma injury, inflammatory injury, or even neuronal injury, apart from the recovery from the injury, so the tissue recover, the pain persists. So this is the problem in terms of chronic pain. When the pain persists beyond the recovery from the disease, the original disease. So very quickly, probably you are aware that pain, there is a pain pathway. So there is special nociceptive neurons that recognize this kind of noxious stimuli in the periphery. We call nociceptors. And these nociceptors, they are very special neurons because they are pseudo-unipolar neurons that has a long axons that innervate the tissue. And they have a cell body in the dorsal root ganglion. And they made up first synapses in the dorsal horn of the spinal cord. And these stimuli can grow until reach the somatosensory cortex, and where the pain is there is a perception of pain in the cortex. So what happens is that when you have dysfunction in this pathway, we may have chronic pain establishment. So what happens is that there is a sensitization, there is a plasticity occurring in this pathway that can lead to pathological pain conditions. So for a long period of time, researchers were mainly concentrating to understand this neuronal plasticity, so especially regarding the plasticity that occurs in these primary sensory neurons, in this nociceptor. But what we know so far in the last decades is that the plasticity of the sensory neurons are not so intrinsic. So there is other cells, other immune cells, glial cells, and resident cells also participate in this process, driving the plasticity of the sensory neurons. So for example, in the dorsal root ganglion, we can identify these very special cells that we call satellite glial cells that change their function during pathological conditions, and they can, for example, they can produce inflammatory mediators that can sensitize the neurons. Or even in the spinal cord, we can describe, for example, the special role of microglia and astrocytes, as Quintana said before, and these cells also participate in the pathophysiology of chronic pain, producing inflammatory mediators that can participate in the plasticity of this pathway that can, in last instance, drives the development of chronic pain conditions. And in terms of what kind of pain our lab is, I'm sorry, I don't know what happens here. In terms of what kind of, I'm sorry, I don't know. What kind of pain my lab is interested? We are very interested to understand the pathophysiology of neuropathic pain. So neuropathic pain is a chronic pain caused by the lesion or disease of the sensory neuron. So when you have a peripheral neuropathy or a central neuropathy, central nervous system neuropathy, we may develop neuropathic pain. So there is several events that can drive neuropathy and can cause damage to the neurons in the periphery, in the central nervous system. For example, autoimmune disease like multiple sclerosis, as mentioned by Quintana previously. We have also metabolic disease that can cause neuropathic pain, such as diabetes. Neuropathic pain can be caused as well by chemicals like cancer chemotherapy. For example, people that are treated with paclitaxel or other chemotherapy drugs, by infection, but also by physical injury. So when you have a compression of neurons, these persons they can develop a neuropathic pain. So my lab, of course, normally use experimental models of neuropathic pain, and we usually different models that are very established in the literature. So the most characterized model in the literature is this model here called spare nerve injury, that we are saying now SNI, in which we cut the peroneal and tibial nerve of the which are branches of the sciatic nerve. And when you analyze the pain hypersensitivity in mouse or even in rats, these animals develop especially mechanical hypersensitivity, which is an important marker of the development of neuropathic pain. We can do this analysis by using different tests, behavior tests, and the most used test is the von Frey filament. So it can evaluate how this injury to the peripheral nerve can induce chronic pain state. So as I mentioned, when we have this kind of lesion in the periphery, several changes occurring across the pain pathway, especially at the local of nerve injury. So there is an inflammation, there is the release of different kinds of inflammatory mediators in this in the site of nerve injury. But there is also neuroinflammation occurring at the level of spinal cord, especially produced by microglia and astrocytes activation. But our lab is mainly focused on the neuroinflammation that is occurring at the level of the dorsal root ganglia. So when you cause this kind of trauma in the periphery, there is also several neuroinflammatory processes occurring at the level of the dorsal root ganglia. And our lab is mainly focused on these mechanisms occurring at the DRG level. So one important process that we are very interested to understand is the interaction, the close interaction between the cell body of the nociceptors that you can see here in purple, and the resident cells, especially macrophages that are resident in the dorsal root ganglion. So you can see in this double staining immunofluorescence that there is a lot of resident macrophages in the DRGs, and these macrophages are in very close contact with the cell body of the sensory neurons. So the question of our lab is to understand the physiological and the pathological interactions between these two cells. And what we know so far is that after this kind of injury, after SNI induction in the periphery, we can induce pain hypersensitivity. And what we found, and not only our lab, but other labs, is that after this injury in the periphery, we can see that there is an increase in the expression of markers of activation of macrophages in the DRG. For example, there is an increase in the expression of IBA1, which is a marker of activation of these macrophages. And there is an increase, also an increase in the number of these macrophages in the DRG. So the idea here is to show you how this peripheral injury can cause an increase in these macrophages in the DRG, and how and what's the mechanisms involved in this induction of this increase in the number of macrophages. So probably you are aware that we have so far, very briefly, we can have two main populations of macrophages as resident macrophages. For example, in the central nervous system, we have microglia as an example of these resident macrophages, and we have the blood monocytes that can be recruited when we have inflammatory process, for example. So how you can distinguish these two populations of macrophages? So one way to distinguish is that normally these resident macrophages, they can express one marker, especially CXCR1 receptor, the receptor for fractalkine. And in the peripheral blood macrophages, they express mainly CCR2, which is could be a very specific marker. So when we want to dissociate which kind of macrophages are important for this increase we have seen before, we can use these both markers of macrophages. So what we did, we breed two strains of mouse. We breed CXCR1 GFP animals with CCR2 RFP animals, and you obtain these heterozygous animals. And you asked which kind of macrophage, resident or blood peripheral, are important for that increase in the number of macrophages after SNI induction. And what we saw is that when we observed a huge increase in the number of CXCR1 positive cells after the injury, but we didn't see any increase in CCR2 positive cells in the DRG, which suggested for us that probably after SNI induction, there is an expansion of CXCR1 cells in the DRG after the SNI induction, after peripheral nerve injury induction, instead of recruitment of monocytes from the blood. To confirm this data, we use another strategy, use a very powerful technique we call parabiosis. So you put two mouse together, so these two mouse can share the blood, and we put we induce the parabiosis in a EGFP animal with a wild type animal. So these animals has all the cells in the blood, all leukocytes in the blood are green. So when the parabiosis is established, these mouse start to share the cells in the blood. So after a couple of days, 30 days and 45 days, we induce SNI in the wild type mouse, and after 7 days, we collect blood and also DRG to analyze if there is a recruitment of cell or peripheral cells to the DRG. As you can see here, there is GFP cells circulating in the blood of the wild type mice, suggesting that our parabiosis were induced. So when you analyze the infiltration of cells in the DRG, we can see that in the DRG of a wild type mice, we saw an increase in the IBA1 cells, suggesting that there is an expansion of macrophages in the DRG, but we didn't observe a significant increase in the number of GFP cells in the DRG, suggesting again that probably the resident cells are proliferating in the DRG after the SNI, which account for the increase in macrophages in the DRG. So to confirm that these cells, these resident cells are proliferating in the DRG, we perform immunostaining for Ki67, which is a marker for cells proliferation. And as you can see here in this immunostaining, we can observe very significant increase in the Ki67 staining in CXT-CR1 positive cells, suggesting that these macrophages are really proliferating in the DRG after peripheral nerve injury. So the next question of our study was, what is the mechanism by which these macrophages are driving neuropathic pain? So they are important for neuropathic pain, and there are evidence that maybe they could produce cytokines after peripheral nerve injury. So we focus mainly on IL-1 beta, which is a cytokine produced by activated macrophages. And as you can see, after SNI induction, there is a upregulation of IL-1 beta in DRG. And when we took advantage of single-cell RNA-seq from public data, we used the single-cell data from this paper just published from Nature Communication by Valéria Cavalli lab. We observed that the most important cell producing IL-1 beta were macrophages, as you can see here. And we also validate this data using cell sorting. So after SNI, we sorting macrophages in the DRGs. And as you can see here, the most important cells expressing IL-1 beta after SNI were really macrophages. So the first part of my talk, we were able to show that after peripheral nerve injury, there is activation and proliferation of resident macrophages in the DRG, and these macrophages start to produce pro-inflammatory cytokines, especially IL-1 beta, and these IL-1 beta are important to maintain and to induce, to promote neuronal hypersensitivity that is very important for the induction of neuropathic pain. And then, what's the next step? What our lab is trying to understand? How this very distal injury in the sciatic nerve can cause this process, this neuroinflammatory process. How this distance injury can lead to proliferation and activation of macrophages in sensory ganglia. So one mediator that our lab is interested in the last 10 years is the complement system, especially 𝗓𝗏𝗄. So we demonstrate that 𝗓𝗏𝗄 is important for inflammatory pain. In this context, 𝗓𝗏𝗄, there is some evidence that 𝗓𝗏𝗄 can drive the infiltration of neutrophils. There is also some evidence in the literature showing that 𝗓𝗏𝗄 receptor must be expressed by sensory neurons. So we took this advantage that we know before, and we start to analyze the importance of 𝗓𝗏𝗄 in the context of neuropathic pain. So firstly, we were able to show that 𝗓𝗏𝗄 receptor knockout animals, they are resistant to the induction of neuropathic pain. So as you can see here, the wild type mice, they develop neuropathic pain, here measured by mechanical allodynia, and these knockouts for 𝗓𝗏𝗄 receptor, they are resistant to neuropathic pain. And then we focus on the DRGs, and we saw that 𝗓𝗏𝗄 receptor is upregulated in the DRG after peripheral nerve injury. And when we analyze it, what kind of cells these increase occur in the DRG, you also use a single cell data. We are able to show that, again, that 𝗓𝗏𝗄 receptor is, even in naive condition, is mainly expressed in macrophages subpopulation, and there is an increase in the expression, especially on macrophages in the DRG. When you decide to confirm this data by immunofluorescence, as you can see here, 𝗓𝗏𝗄 receptor is expressed in cells express IBA1, which is a marker of macrophages. And we were also able to show that transgenic animal that express 𝗕𝗏P under the control of promoter of 𝗓𝗏𝗄, we saw that macrophages are really the source and are really the cells expressing 𝗓𝗏𝗄 receptor in the DRG. And also in human DRG, so we took advantage and collaborate with 𝗕𝗏𝗏 from Cincinnati University, and we were able to show that 𝗓𝗏𝗄 receptor is really expressed in human DRG. And when we analyze by immunofluorescence the expression of 𝗓𝗏𝗄 receptor, we were able to show that 𝗓𝗏𝗄 in the human DRG is expressed by cells expressing IBA1, suggesting that even in humans, DRG 𝗓𝗏𝗄 receptor type 1 is expressed in macrophages. So in attempt to confirm that our phenotype in the knockout, in full knockouts of 𝗓𝗏𝗄 receptor is due to a mechanism occurring in the DRG macrophages, we used conditional animals. We produced conditional animals in which we knock out 𝗓𝗏𝗄 receptor only in myeloid cells using lysem cream animal, which is expressed, which we know here that lysem is expressed by these macrophages in the DRG. So we knock out 𝗓𝗏𝗄 receptor only in this macrophage, but also in peripheral macrophages. But we rule out the role in peripheral macrophages. And we observed that these animals are also resistant to the development of neuropathic pain, indicating that probably these macrophages, these DRG macrophages, are important for neuropathic pain through activation by 𝗓𝗏𝗄 receptor. And what's the mechanism by which 𝗓𝗏𝗄 receptor is driving the activation of macrophages and contributing to neuropathic pain? So we showed that mainly 𝗓𝗏𝗄 receptor is important to drive the production of IL-1 beta, which is the mediator that we described before, being important for neuropathic pain development. And then, of course, as I mentioned in the beginning of my talk, our interest is to develop novel therapeutic strategy to treat this disease. And we have collaborated with 𝗕𝗏𝗄, which is a company in Italy, in the development of a novel antagonist of C5a receptor, which is a very potent and selective molecule, DF2953, which is by docking, we were able to show that binds to C5a receptor before. And we tested these antagonists in our model, and as you can see, these antagonists were able to block and to reduce neuropathic pain. So in the last instance, our take-home message is that there is neuroimmune interactions in the DRG, and these neuroimmune interactions is driven by complement system through activation of macrophages. And we are proposing now that these macrophages and this pathway is an important target for neuropathic pain control. So I'd like to thank my students and postdocs in the lab. Our first meeting still 2019, our last party at the end of the year. I'd like to thank our collaborators, Fernando, Zeca, and also people from 𝗕𝗏𝗄 and Timo from Cincinnati University, and our founding. And thank you very much for your attention. I would like to answer any question that you may have.
Okay, Thiago, great talk. We have already a number of questions for you, of course. Let me see if you can, if there's enough time for you to answer to a few of them. So first, I hope I get the right sequence. Sérgio Ferreira asked whether the number of CX3-CR1 positive cells appears to decrease in the DRG from 1 to 2 weeks after the injury. So I believe he wants to comment.
Yeah, what we saw is that there is an expansion of these cells that is in parallel with the induction of the neuropathic pain condition. But after 2 weeks, 14 days, 24 days, there is a reduction. There is a reduction in the number of these cells. We don't know the mechanism by these cells retract, for example, if there is an apoptosis of these cells. But we saw is that although there is a reduction in the macrophage number, we still have pain hypersensitivity. So it seems that these macrophages must be mainly important in the induction of the process, in the induction of neuropathic pain. But of course, although we have a reduction in the number of these cells, we don't know exactly if these cells still producing some mediators, still producing molecules that can maintain the neuropathic pain condition. So this is an important question that we don't have the answer yet. So we are very interested to understand.
Okay. Thank you. Have you already looked at in the spinal cord with your dual fluorescent animals to see if there too is mostly proliferation of resident cells instead of infiltration?
Yes, it's a great question. Yes, the answer is yes. We have analyzed the spinal cord, and of course there is a proliferation of spinal cord microglia. But we never, at least in this model that we have used, the SNI model, we never saw infiltration of monocytes. And we have published this data already 2 years ago in the Journal of Leukocyte Biology. And other labs also confirmed this data. And because there is previous papers showing that maybe monocytes can infiltrate the spinal cord, but what we found is that this paper, there is an artifact in that paper that they use irradiation as a process to repopulate cells. So we know that when you have irradiations cells from the periphery, they can go into the spinal cord. So there is a bias in this paper. So now it's the people in the field are very confident that cells are not able to infiltrate the spinal cord after SNI or other peripheral nerve injury. It's fine.
Okay. So a related question, 𝗕𝗀𝗪𝗪 asked whether at YouTube, do you think other immune cells can invade the DRG and also contribute to the development of neuropathic pain, as lymphocytes and neutrophils?
So it's another important question. Of course, we focus on the macrophages, but our parabiosis data, it's very clear that there is not too much cells infiltrating, at least in the DRG. We don't know why, because there is immune mediators being produced in the DRG, so we don't know why these cells can't infiltrate the DRG. But we have data that we just published, it's not published, it's in the bioarchives, in which we saw that these cells, although these cells are not able to invade the parenchyma of the DRG, they stuck in the leptomeninges that surround the DRG. So we are very interested to analyze how these cells infiltrate in the leptomeninges and why they can't infiltrate the DRG. So we are now investing a lot of time to understand why these cells, and there we have a lot of different cells infiltrating. Lymphocytes, dendritic cells, neutrophils, monocytes. So we are doing single cell sequencing now to see what kind of cells are accumulating. We don't know if they are infiltrating or from the blood, but they are accumulating in the leptomeninges of the DRGs.
Okay, so I have to apologize to Júlio Schaffstein, Marília, Lucas Bolini, and others. We will put the questions, we'll forward the questions to Thiago and post it at YouTube, okay, the answers, so that you can see, so that we can move on and not be too much delayed. So Thiago, thank you very much for the beautiful talk. Let's move to our third presenter this morning, Professor Alexandre Basso. Is Professor of Microbiology and Immunology at the Federal University of São Paulo School of Medicine. And he'll talk about his past contributions have been both in the central nervous system, like Professor Quintana's, and also in peripheral neuroimmune interactions, a bit like what Thiago is doing. So Professor Basso sought to put together brain, the medulla, and the periphery together. So he's had interesting contributions in the role of the neurosystem in inflammatory processes such as allergy in the lung, and also is interested in multiple sclerosis and the models of encephalomyelitis, which I think are the focus of his talk today. So the room is yours. Professor Basso, thank you very much for coming.
Thank you, Newton, for the kind introduction. I also would like to thank the organizing committee for the invitation to be here today. And I'm honored actually to share the section with all these distinguished speakers and very good friends. So the lab has been interested in the crosstalk that takes place between the nervous and the immune system. So as you know, in one direction, the nervous system, by releasing neurotransmitters and neuropeptides, can modulate immune cell function and thus control immune and inflammatory responses. And there are at least 4 very known pathways operating this kind of interaction. So the sympathetic fibers, HPA axis, sensory fibers, and the vagus nerve. And in the other direction, it is also true that by releasing cytokines, chemokines, and other inflammatory mediators, immune cells can impact the function of the central nervous system, so modulating the activity of specific brain areas and impacting, for instance, mood and behavior. In the past few years, the lab has been studying how the sympathetic nervous system can impact or can influence the development of autoimmune disease, and in particular I will talk about the MS model in animals that Francisco already introduced, which is EAE. So the sympathetic nervous system is one of the divisions of the autonomic nervous system, and although we don't see any lymphoid organ listed among those receiving sympathetic fibers, we do know that indeed lymphoid organs are also innervated by the sympathetic nervous system. So that is true for primary lymphoid organs such as the thymus and the bone marrow, and this is also true for secondary lymphoid organs such as the spleen and lymph nodes. And then the question is, what are those fibers doing there? So is this any relevant in order to modulate the activity of the immune system and neuroinflammation? So most of the studies trying to address these questions in vivo, they have been using, they have been employing a technique that is related to the ablation of sympathetic fibers by treating animals with 6-hydroxydopamine. And the reasons that I think it's not worth mentioning today, we have been using a different approach that I will try to explain to you right now. So here we are looking at a sympathetic fiber. So we have the noradrenaline being produced here, and once the noradrenaline is released into the synaptic space, of course it can bind to postsynaptic receptors, but it can also bind to presynaptic receptors, and those are alpha-2A, alpha-2C adrenergic receptors. And what is the purpose of noradrenaline signaling through these presynaptic receptors? So we know that the activation of these alpha-2A, alpha-2C adrenergic receptors decreases the production of cyclic AMP, leading to an inhibition of further release of noradrenaline from the neuron. So this is actually an important negative feedback mechanism controlling the activity of the sympathetic nervous system. So animals that are double deficient to alpha-2A, alpha-2C adrenergic receptors, they miss this negative feedback, and because of that, they are characterized by hyperactivity of the sympathetic nervous system. So we can see here that those double deficient alpha-2A, alpha-2C knockout mice, they have increased concentrations of noradrenaline in the bloodstream as compared to single knockouts and wild-type mice. So we have been using these animals as a model to understand how the sympathetic nervous system can regulate or control the development of EAE. So here we can see that the animals with a hyperactive sympathetic nervous system, they actually develop less severe EAE as compared to wild-type animals. So we didn't see much of a difference in the incidence of the disease, but we can clearly see a huge difference in the maximum score, meaning the peak of the disease, and actually we can appreciate a huge difference along the disease course after the peak of the disease. We also decided to look what was going on inside the CNS the inflammatory response inside the CNS. So here we are looking at the mononuclear cells infiltrating the CNS at the peak of the disease. So CD45 and CD11B, and what we found was that the 𝗨𝗂𝗂𝐴, 𝗨𝗂𝐴 deficient animals, they had a reduced in the frequency of this population here. So those are activated microglia and infiltrating macrophages or monocytes. And on the other hand, an increase in the frequency of resting microglia, as we can appreciate here. And actually, the total number of CD11B, CD45 high cells infiltrating the CNS is also lower in 𝗨𝗂𝗂𝗏, 𝗨𝗂𝗂𝐶 deficient animals. I'm talking about this population here. So again, mostly activated microglia or infiltrating monocytes or macrophages. And regarding the T cells, so here this is showing interferon gamma and IL-17A expression in CD3, CD4 positive T cells. So we also found a reduction in the frequency of interferon gamma expressing CD4 T cells and a reduction in the expression of interferon gamma by those CD4 T cells infiltrating the CNS at the peak of the disease. And the total number of CD4 T cells infiltrating the CNS is also reduced in the animals with the hyperactive sympathetic nervous system. And we didn't find much of a difference in the frequency or in the expression of IL-17 in CD4 T cells. So the bottom line here is that along with less severe EAE, the animals also has a less inflamed CNS at the peak of the disease. But that was about the effector phase of the disease. We wanted to know whether or not or how was the generation of the adaptive effector T cell responses that are responsible for EAE. So in order to interrogate that, we immunized animals with MOG3555 in CFA, and 7 days later we took the cells from the draining lymph node and we cultured the cells in vitro with antigen to check for antigen-specific induced cytokine production and proliferation. And as you can see here, the cells collected from the animals with a hyperactive sympathetic nervous system upon antigen-specific stimulation, they proliferated at lower rates, and also they produced lower amounts of IL-2, interferon gamma, and very importantly, lower amounts of GM-CSF alpha. And once again, we didn't find any difference in IL-17A. So less severe EAE in those animals is associated with an impaired generation of the effector adaptive immune response. So the encephalotogenic immune response that is responsible for EAE development in those animals. So I don't have time today to show you the whole data, but we had good reasons to believe that at least in part, the increased activation of beta-2 adrenergic receptors could explain at least in part the phenotype, the EAE phenotype in those animals with the hyperactive sympathetic nervous system. And the beta-2 adrenergic receptor is the most expressed adrenergic receptors in immune cells, including CD4 T cells. And the signaling pathway downstream beta-2 engagement involves adenylyl cyclase conversion of ATP into cyclic AMP, activation of pKa, and then Krebs phosphorylation. And this is exactly what we found in CD4 T cells. So here you can appreciate expression of beta-2AR on the surface of CD4 T cells, naive CD4 T cells, sorted naive CD4 T cells. And upon activation of beta-2AR in these cells, what we found was increased Krebs phosphorylation that occurs in a pKa-dependent manner. So when we block it, pKa using this peptide here, we found a decrease in Krebs phosphorylation upon treatment of these cells with beta-2AR agonist. So this is an indication of increased intracellular levels of cyclic AMP upon beta-2AR activation in these cells. And we know that from the literature that increased cyclic AMP levels inhibit T cell activation and proliferation through at least two different described mechanisms. So again, this is exactly what we found in our case. So here we are looking at a proliferation assay in which we had LPS activated dendritic cells posted with MOG3555, and they are presenting antigens to 2D2 T cells. So those are T cells expressing a transgenic TCR recognizing MOG3555. And we had here in this assay non-treated 2D2 T cells or 2D2 T cells treated with an agonist of the beta-2 adrenergic receptors. And those are here in blue. And we can see that the 2D2 T cells that had been treated with the beta-2AR agonist, they proliferated at lower rates as compared to non-treated 2D2 T cells. So beta-2AR signaling indeed inhibits CD4 T cell proliferation upon antigen-specific stimulation. And so if that is the case, what if we block beta-2AR signaling in the animals with the hyperactive sympathetic nervous system? So is this enough to recover the EAE phenotype? So we set up this experiment here in which we had wild type animals, and once again the alpha-2A, alpha-2C deficient animals, in which we implanted osmotic pumps delivering either vehicle or an antagonist of the beta-2 adrenergic receptor. And that was done during the generation of the adaptive immune response. So later on, after disease onset, we didn't have any more the antagonists being delivered to these animals. And as you can see here, the pharmacological blockade was enough to recover EAE development in these animals. So once again, we can see here that the alpha-2A, alpha-2C deficient animals, they develop less severe EAE. But after the pharmacological blockade during the generation of the adaptive immune responses, these animals with the hyperactive sympathetic nervous system, they develop very similar EAE as compared to wild type animals. And in addition, the pharmacological blockade of the beta-2 adrenergic receptor was also sufficient to recover or to avoid the impairment in the generation of the adaptive immune response. So once again, when we took the cells from the draining lymph nodes of the animals with the hyperactive sympathetic nervous system, we could see that upon MOG3555 stimulation, they produced lower amounts of GM-CSF and interferon gamma. And after the pharmacological blockade, those cells were producing GM-CSF and interferon gamma as much as did the cells coming from the wild type animals. And once again, we didn't find much of a difference in IL-17A production. So the bottom line here is that treatment of alpha-2A, alpha-2C deficient animals with an antagonist of the beta-2 adrenergic receptor, reverses less severe 𝗂𝗂 and the impaired generation of the adaptive immune response. And we also did some experiments with animals that are knockout of the beta-2 adrenergic receptor. In this case, we saw just the opposite. So the animals that are deficient in the beta-2 adrenergic receptor, they accordingly, they develop more severe 𝗂𝗂, in an indication that even in normal status of activity, the sympathetic nervous system via signaling through the beta-2 adrenergic receptor is likely to regulate the development of 𝗂𝗂. But we wanted to know whether or not beta-2 signaling specific immune cells would be important for the sympathetic nervous system regulation of 𝗂𝗂 development. So to interrogate that, we did these chimera experiments in which we had irradiated wild type as recipients being reconstituted with either bone marrow cells coming from wild type congenic animals or congenic animals that are deficient for the beta-2 adrenergic receptors. And we can see here that the lack of beta-2 only immune cells recapitulates the 𝗂𝗂 phenotype of the entire knockout. So they develop more severe 𝗂𝗂. And talking about the generation of the adaptive immune response in those animals, we can see here that upon antigen-specific stimulation, the cells coming from the draining lymph nodes of the recipients reconstituted with the beta-2 deficient cells, they produced higher amounts of interferon gamma, GM-CSF, and also IL-2. So finally, we asked whether or not beta-2 signaling specifically in CD4 T cells would be important to the sympathetic nervous system regulation of 𝗂𝗂 development. So in this case, we set up this experiment here in which we had wild type 2D2 cells being transferred either to REG1 knockout animals, so REG1 knockout animals with a normal sympathetic nervous system, and REG1 knockout animals with a hyperactive sympathetic nervous system. And along with that, we also had 2D2 cells that were deficient for the beta-2 also being transferred to REG1 knockout animals with a hyperactive sympathetic nervous system. And what we saw here was that the 2D2 cells, they induced less severe 𝗂𝗂 when they were transferred to animals with a hyperactive sympathetic nervous system. Okay, but then if now we transferred 2D2 cells lacking the beta-2 to the very same animal, to the animals with a hyperactive sympathetic nervous system, now they are able to induce similar disease as the wild type 2D2 cells can induce when they are transferred to animals with a normal sympathetic nervous system. And also, when we took the cells from the central nervous system at the peak of the disease, we can see here once again that the cells coming from the recipients with a hyperactive sympathetic nervous system, upon antigen-specific stimulation, they secreted lower amounts of interferon gamma and GM-CSF. But once again, when we now transfer 2D2 cells lacking, that are deficient for the beta-2 adrenergic receptors, they are back able to produce high amounts of interferon gamma and GM-CSF. So the conclusion of this part of the talk is that the sympathetic nervous system hyperactivity can modulate the adaptive immune responses, mitigating autoimmune CNS inflammation. And sympathetic nervous system regulation of immune cells most likely relies on beta-2-ER signaling, limiting the generation of the encephalitogentic TCR response. And this is the graphical abstract. I didn't have time to show you all the data today, but we saw that likely beta-2-ER signaling is increasing 𝗬𝗓 expression, and this increased 𝗬𝗓 expression is associated with the impairment in the generation of the encephalitogentic TCR responses with reduced interferon gamma, IL-2, and GM-CSF production upon antigen-specific stimulation. So I already mentioned that in the beginning of my talk, this crosstalk between the nervous and the immune system, but it is actually much more complex than that. And actually, the nervous and the immune systems, they share a common language. And in this sense, the neurons can also express and secrete cytokines and chemokines. And at the same time, immune cells are shown to secrete neurotransmitters and neuropeptides in such a way that now one can play the role of the other in this kind of interaction. So here we can see a PNS paper, actually an old one, showing that sympathetic neurons can produce and secrete IL-6. And this other one in JCI, it's a very nice paper in which the authors show that parasympathetic neurons can express and secrete eotaxin. So here we can see in the airways. So this is in the airways, and parasympathetic fibers expressing eotaxin, and those are immunofluorescence showing neurons expressing eotaxin. And what they show, it's a very nice paper showing that IL-4 driven eotaxin released by nerve fibers can attract to eosinophils to the airways. And once the eosinophils are activated, they release major basic protein, and this will inhibit M2 receptor function, leading to increased release of acetylcholine and increased bronchoconstriction in animal models of lung allergic inflammation. And as I said, this is also true for immune cells. So it has been shown that lymphocytes can indeed express all the enzymes that are necessary for them to secrete neurotransmitters as catecholamines and also acetylcholine, and in particular, human regulatory T cells selectively express tyrosine hydroxylase and are able to secrete catecholamines. But there's still very little knowledge on how important this is, especially in vivo. So we decided to address this question using conditional knockouts in which only FOXP3 expressing cells are deficient of tyrosine hydroxylase. So the tyrosine hydroxylase is the rate limiting enzyme in the noradrenaline pathway synthesis. So here we decided to use FOXP3 YFP mice bred with pH fluxed mice. So in a way that now we have only the FOXP3 expressing cells missing pH expression. So, and here we can see that indeed FOXP3 regulatory T cells express tyrosine hydroxylase. So those are sorted regulatory FOXP3 expressing T cells. And of course, that as compared to the central nervous system, they produce lower amounts of tyrosine hydroxylase, but it is still there. And here, and also dopa beta hydroxylase, which is the enzyme doing the last step, so converting dopamine into noradrenaline. And here we are showing that indeed the 𝗕𝗏-FOXP3 the FOXP3 th flox mice, they do not express th, and there is also a reduction in the expression of dopamine dopa beta hydroxylase in those animals. But what about the secretion of noradrenaline in those animals? So we use mass spectrometry to analyze noradrenaline secretion by FOXP3 expressing regulatory T cells sorted from the spleen and lymph nodes of the animals. And we can see here that the Treg cells are able to secrete noradrenaline, and naive T cells that do not express FOXP3 secrete very low amounts of noradrenaline. And once again, we can see here that after ablation of the TH using the FOXP3 animals, they are no longer able to secrete noradrenaline. Okay, so but what was the consequence for that in terms of the suppressive function of the FOXP3 expressing regulatory T cells, FOXP3 positive regulatory T cells? So here we are looking at data on a suppression assay in which we had naive wild type responder T cells cultured along with either control FOXP3 positive cells or TH deficient FOXP3 regulatory T cells in black. And we can see here that the responder T cells that were cultured along with the TH deficient FOXP3 regulatory T cells, they proliferated at higher rates as compared to the ones cultured with the control FOXP3 regulatory T cells. So indicating that the TH deficient FOXP3 regulatory T cells display reduced suppressive function. Interestingly, this was true only if the naive T cells express the beta-2 adrenergic receptor. So here we can see that when we did the very same experiments in naive T cells lacking the beta-2 adrenergic receptors, this was no longer true. So there was no difference in the suppressive function comparing the two FOXP3 expressing T cells. So the hypothesis that we are drawing here is that maybe the secretion of noradrenaline by FOXP3 regulatory T cells may be an additional mechanism of suppression used by these cells. And what about in vitro? So looking at the generation of the encephalotogenic T cell responses, comparing those two animals, we by doing ELISpot, we found a difference in the number of interferon gamma producing cells. So the animals with FOXP3 T cells that are deficient for TH, they had a higher number of interferon gamma producing T cells, but we went to check the amount of cytokine produced by cells collected from the draining lymph nodes 7 days after immunization. We can see here that upon antigen-specific stimulation, we couldn't find any difference in IL-17 or in interferon gamma, but we did find higher amounts of GM-CSF production by those animals. And the bottom line, when we did the in vivo EAE experiment, we also found that the animals that are deficient of the tyrosine hydroxylase only in the FOXP3 expressing regulatory T cells, they develop more severe EAE, and the maximum score is higher. and also we can find difference all along the curve. So the regulatory T cells are very important for this partial remission phase that the animals display here, and we have to look better what's going on here. We just look at the degeneration of the adaptive immune response, as I just showed you, but we didn't look in detail what's going on here in those animals. So to conclude this last part of the talk, it seems that the secretion of noradrenaline may be an additional mechanism of suppression in 𝗏𝑥P3 regulatory T cells. And by releasing noradrenaline, 𝗏𝑥P3 regulatory T cells occasionally play the role of the sympathetic nervous system regulating immune response. So I will stop here, acknowledging people who did the hard work in the lab. The first part of the talk was done by Leandro Pires Araújo, a former PhD student and postdoc in the lab. He's now at Columbia University doing another postdoc. And the second part of the talk was done by Márcia Guereschi, and they had help from many other people in the lab. I also would like to acknowledge the collaborations with Patrícia Brum from USP and Francisco Quintana and the funding agencies. So thank you very much for your attention, and I can try to answer questions if we still have time. Thank you very much.
Oh, Alexandre, thank you very much. Beautiful talk, very interesting models, and we have very interesting questions too, but we are short of time. So I will ask you to answer just the first question live, and then perhaps if you can, we'll post your replies in the YouTube chat, okay? So Lucas Gonçalves asked whether your data, especially regarding IL-17 and GM-CSF, suggest a very likely role of ILC3 on EAE development. Have you assessed this population?
No, we have not assessed this population. I have to say that in these assays, all the produced cytokine that we measure in these ex vivo experiments were after antigen-specific stimulation. So I don't think that the cytokines would be coming from ILC3 in this case, but it is a very interesting question. I mean, there is data from David Artie's lab showing that beta-2-AR signaling in ILC2 can modulate the function of these cells in helminthic infections, and maybe this is happening also in ILC3 cells, but we didn't look at that. It's a very interesting question.
Okay. Well, thank you very much, Alexandre. I think we need now to thank you for the participation. We'll have your answers in the chat. I'd like to invite Josiane, who has some announcements to make, and then we'll follow for the next section.
Okay, thank you very much.
We thank you.
Hello, everyone. So just before we go on, quick announcement, actually. Just invite everybody to, on Monday, next Monday, October 18th, we're going to have, after our regular section on microbiota at 10 a.m., we're going to have in the afternoon a special section about where our students and postdocs will be presenting their work. So I'd like to invite and remind everybody to be there, to ask questions and to appreciate the works. Also, I'd like to remember that all the abstracts are available at our website. So all the abstracts and also the nominated ones. So go there and take a look. So I'm gonna call now Gilda Neves that's gonna introduce our next speaker. Thank you.
So thank you, Josiane. Good morning, everyone. So it's my pleasure to introduce you the final speaker of this morning. That is Professor 𝗏𝘂𝘶-𝗵𝗏. Professor 𝗧𝗵𝗵𝗃𝗏 is a full professor at the Biophysics Department at Federal University of Rio de Janeiro. He is definitely a leading researcher in the field of neurobiology, in the study of the neurobiology of neurodegenerative diseases. He has several national and international prizes and more than 150 highly cited papers in the field, mostly of the neurobiology of Alzheimer's disease. And more recently, his group have been making several contributions for the understanding of the neuroinflammatory responses after viral infections, especially a virus that has some kind of neurotrophy. So please, Professor Sergio, it's my pleasure, and the screen is yours.
Thank you very much, Gilda, for the nice introduction. And let me place my talk here. And I would, of course, like to begin by thanking you, Gilda, for the introduction, Claudia for the invitation, and all the organizers of this meeting for the opportunity to present some of our work here. It is definitely a great pleasure. It's been a very exciting meeting. And I think that speaking after the 3 previous speakers today is going to be a hard act to follow for sure. But I hope that I can keep you from running out to lunch for the next 20 minutes or so. So the story that I would like to talk to you about today began, as in most cases with COVID research or SARS-CoV-2 research, about 1.5 years ago, when the pandemic actually reached Brazil and was already spreading globally and causing large numbers of infections and deaths. And so I think that as many other people, as probably most people in the world, we started considering what kind of impact the pandemic would have on our lives, our work, our health, of course. And it was kind of inevitable to begin thinking about how the pandemic might also affect our subject of research, the central nervous system, CNS. So led by Fernanda de Felice and in collaboration with some other colleagues from Brazil and Canada, we published a perspective article back in April of last year. So as I said, about 1.5 years ago, dealing with the possible consequences of infection by SARS-CoV-2, the causative agent of COVID-19, on the central nervous system. And one of the things that we pointed out at that early point in time in the pandemic was that most likely it would be necessary to perform detailed and longitudinal studies, including brain imaging, neurological and neuropsychological evaluation, to really examine multiple cognitive domains and other neurological functions in patients that had and survived COVID-19, and to see how that having had the disease might affect their neurological system. So how, at this point, I should probably make a disclaimer. As Gilda mentioned, I'm neither a virologist nor an immunologist. Instead, I'm a neuroscientist who studies Alzheimer's disease. And so I thought I would, in order to tell you how I ended up doing some work related to brain inflammation in viral infection and now with COVID-19, I thought I should guide you through a brief story of what we've been doing in terms of brain inflammation in the context of Alzheimer's disorder. So the main topic of my lab has been on the investigation of these oligomers, these small aggregates formed by this peptide beta amyloid, and these oligomers attack neurons. And many labs around the world, including my own lab, have shown that oligomers, they trigger a number of key pathological features of Alzheimer's disease, including tau hyperphosphorylation, brain oxidative stress, which are really hallmarks of AD neuropathology. They trigger brain insulin resistance, making AD, Alzheimer's disease, a sort of a brain diabetes, if you will. But of more importance today, oligomers trigger brain inflammation, the loss of synapses, and because of the loss and functional impairment of synapses, that leads to memory impairment. So I would briefly show you some examples of our own findings of how brain inflammation occurs in Alzheimer's and how that's connected to the accumulation of these toxins, A-beta oligomers that accumulate in the Alzheimer brain. So here are some immunohistochemical analysis of IBA1 positive cells and GFAP positive cells in the brains of mice that received an intracerebroventricular administration or infusion of A-beta oligomers. And when we counted the number of IBA1 positive cells or microglia or myeloid cells, we saw a marked increase in the numbers of cells in the brains of mice that received A-beta oligomers compared to control mice that received vehicle. And the same happened with astrocytes, with GFAP positive cells. And just to give you another example of how dramatic this response is, and now focusing on microglia, which will be more of the topic of my talk today, this is labeling for IBA1 again in the control mouse brain. So you see a few microglia surveilling the dentate gyros of the hippocampus shown here. The cells are labeled in blue with DAPI. And in the brain, in the hippocampus of a mouse that received an infusion of A-beta oligomers, we see a very marked increase or expansion in the number of IBA1 positive cells. And of course, these could be infiltrating macrophages, as we heard from Thiago, for example, but we did label them with TMEM119, which is supposed to be much more specific for microglia, and we see a similar increase in microglia labeled by TMEM119 as well in the brains of oligomer-injected mice. So interestingly, that is not exclusive of rodents. Indeed, in collaboration with Fernanda de Felice again and with Doug Munoz from Canada, who have developed a very interesting non-human primate model of Alzheimer's disease, we showed that inflammation is also very evident in the brains of monkeys that received injections of A-beta oligomers compared to control sham monkeys. So labeling for GFAP here is markedly increased in different regions of the brain, the frontal cortex, the dentate gyros, the amygdala, and the same holds for IBA1. So again, astrogliosis and microgliosis induced by A-beta oligomers also in a primate brain, in a non-human primate brain, in addition to the rodent brain. But of course, most of the work that we've done in the lab has been in mice. And in addition to gliosis, the proliferation of cells of microglia and astrocytes, we've also found that soluble factors like pro-inflammatory cytokines are also increased in the brains of mice that receive infusions of A-beta oligomers. So here's, for example, IL-1 beta and TNF-alpha, and we've also been doing some studies with IL-6. And with IL-1 beta and IL-6, we've done fewer studies, but with TNF-alpha, we've done much more detailed studies. And we found in work, again, by José Henrique Ledo, who was a grad student in the lab and is now a junior faculty, beginning his lab at University of California in Southern California. And Michel Lourenço, who was also a student in the lab and is now an assistant professor at the Institute of Biochemistry at the Federal University. We showed that the increase in TNF-alpha in the brains of these mice is behind the impairment of memory. So there is a activation of a signaling pathway, or multiple signaling pathways, in fact, that culminate with memory impairment in these mice. And it's mostly due or initially triggered by TNF-alpha, and which comes from microglia. Now, in addition to secretion of these soluble pro-inflammatory factors, in the Alzheimer's field, there has been increasing recognition of the fact that microglia may also lead to synaptic damage and to memory impairment by an additional mechanism, namely what is known as synapse pruning. So this is a physiological process that occurs in development. So microglia in the developing brain, they prune or remove phagocytose and remove excess synapses And in Alzheimer's disease, however, it has been shown originally by Ben Barres, also by Bat Stevens, and now by other researchers in the area, that in Alzheimer's disease brains and in Alzheimer's disease models, microglia are triggered to engulf synaptic elements, both pre and postsynaptic terminals, and that is a process that is driven or triggered by the complement system, by C1q and C3 in particular. And we found that actually blocking TNF-alpha, which initiates this cascade, or with a monoclonal antibody that blocks TNF-alpha, or blocking microglial activation with minocycline, or removing microglia by depleting the brain of microglia with sodium chloride, or even blocking proteins of the complement system, all those approaches are capable of rescuing or preventing memory damage induced by A-beta oligomers. So we were quite happy doing our research in Alzheimer's disease a few years ago, 5 or 6 years ago, when all of a sudden Brazil was a target of Zika fever. So you may all remember that a few years ago we had this epidemic of Zika, which fortunately remained more or less restricted to Brazil as an epicenter at that moment, didn't really spread worldwide as was originally thought it might occur. But Zika, of course, is a very complex disorder with many organs and tissues affected, including the brain. For example, patients affected by Zika fever, they have pronounced and strong headaches. And that, of course, is indicative. The fact that many organs, peripheral organs in the CNS are affected is indicative of a relevant role of a cytokine storm in this. And in Brazil, it was found that one of the really most prominent features of Zika infection was microcephaly in babies born from mothers that had been infected by the virus. And several studies, very nice studies from, including nice studies from colleagues in Brazil, show that Zika virus targets neuroprecursor cells, and that kills those cells and prevents brain development, possibly by killing neuroprecursor cells. Now, we were interested at that point in contributing to this effort to understand the neurological impact of Zika virus, and we decided to ask whether, in addition to the marked effect on microcephaly during brain development, if postnatally Zika might also be affecting the CNS. And for that, we first teamed up with a number of colleagues to perform a study on the more acute or more chronic neurological impacts of early life, early postnatal infection by Zika virus in mice. And then what I would like to tell you about today, work that we did in adult mice, showing that actually infection by Zika virus in adult wild-type mice may impair synapses and memory in such mice. And so I just want to give you a brief highlight of a couple of results from that study. This is a labeling for synapses in the brains of mice that have been infected with Zika virus in the brain. So in green is synaptophysin, and in red is HOMR1, and these are pre or post synaptic markers. So the green marks presynaptic axon terminals, the red marks postsynaptic densities, or the dendritic spines that assemble into the synapse. And as you can see here, there's a lot of yellow in the cells, revealing a large number of juxtaposition of pre and postsynaptic terminals, and thus structure of synapses. Now, when the mouse is infected by Zika virus, a few days after infection, we see that there is a marked reduction in the number of yellow dots, showing that there is really a decrease in the number of structural synapses. And this is shown here both in the dentate gyrus and in the CA3 region of the hippocampus, just as examples of the mouse brain. And this was work that was really done by Cláudia Figueiredo and Fernanda Barros Aragão. Fernanda Barros Aragão is now a postdoc in Fernanda de Felici's group, and Cláudia Figueiredo was already and still is a professor at the School of Pharmacy at our university. Now, in addition to synapse damage and memory impairment, which I didn't have time to show, what they found was that there was a really marked increase in the number of IBA1 positive cells. So we see here the hippocampus of a mouse that was infected by Zika virus compared to a control mock infected mouse. So there are a few microglial cells here and a lot of microglial cells here. And interestingly, this large increase in microglial immunoreactivity decreased as a function of time. So at about 2 months post-infection, it was almost non-existent anymore. So the density of microglial immunoreactivity, it was very similar to the control levels. And this kind of matches or coincides in time with the recovery of memory. So mice had memory impairment after about very early on, a few days after infection. This memory impairment persisted for about a month or so when microglia were still high. And then by 2 months, when microglia were already down, memory was recovered. So suggesting that there was a close parallel between microglial reactivity and memory impairment. And one of the things that we also looked at, of course, was the release of and production of inflammatory cytokines. So here I'm actually showing expression, so production of these cytokines in the brains of mice. So we see there is quite an increase in IL-1 beta and a very large increase in TNF-alpha. And indeed, we thought to ask whether this increase in IL-1 beta was relevant to Zika infection. And we found that at least in terms of memory impairment, IL-1 beta does not appear to play a role, because mice, there are knockouts for the IL-1 beta receptor, show the same type of memory impairment induced by Zika virus. On the other hand, we see that there's this tremendous increase in TNF-alpha, about 50-fold increase in TNF-alpha expression in the brains of mice infected with Zika. And so we thought that this might be an important cytokine in memory impairment and synapse damage. And in fact, we found that to be the case. And again, we thought about what we learned about Alzheimer's disease. And in particular, we asked whether microglial mediated pruning of synapses, this engulfment and phagocytosis of synapses, might be taking place in the brains of mice infected by Zika. So, and indeed, we were stimulated by the fact that a couple of years before, there was this nice paper by the group of Robin Klein showing that in infection by West Nile virus, there is a microglial-dependent mechanism that really engulfs and phagocytoses synapses, and that this is triggered by complement and by the activation of complement, as is now thought to be the case in Alzheimer's again. And so we looked at complement in our mice that had been infected with Zika virus, and we looked at both C1q and C3, and we see that in mice infected with Zika virus, there is a very large increase in both proteins of the complement system here, suggesting that indeed microglia might be acting as phagocytic cells and eating up synapses in the brains of mice that were infected. And indeed, when we looked at the confocal microscopy here, staining for synapses with presynaptic markers, synaptophysin, and microglia with IBA1, we see that in mock-infected mice, microglia are there just surveilling the synapses as they normally should. So there are processes here. They're probably surveilling the environment and sensing for a presence of any pathogens or activating molecules. And here, in contrast, when the mice was infected with Zika virus, we found a lot of yellow spots inside microglia, as you can see as these arrowheads here, suggesting that synaptophysin-containing presynaptic terminals had indeed been engulfed by microglia. And when we count these, we see that there is a very large increase here, about twofold increase in the number of synaptophysin immunoreactivity within microglia. So suggesting that a similar process to what goes on chronically in the Alzheimer's disease brains occurs very acutely in the brains of mice infected with Zika virus. A very strong parallel between the two disorders, except that one, of course, is chronic, takes decades to develop in the case of Alzheimer's, and the other one is very acute, as in the case of Zika virus infection. And in the same study, we found that blocking TNF-alpha signaling or blocking microglial activation or blocking complement proteins with antibodies against C1q or C3, we completely prevent neuronal damage and memory loss in mice infected with Zika virus. So that suggests that indeed brain inflammation and microglia play key roles in memory impairment and neuronal damage induced by Zika. So this brings me back to the COVID story that I began my talk with. And in the last few moments, I just want to show you what we've been interested in studying. And after we published this prospective article in April of last year, there have been a number of studies, clinical and pathology studies, trying to determine to what extent the central nervous system is actually targeted by Zika virus. Sorry, by SARS-CoV-2. And this is just one example of a systematic review by Brazilian colleagues who published a number of neurological manifestations, including cerebrovascular disease, encephalopathy, and more benign impacts like anosmia and ageusia, headache, myalgia, and other neurological impacts as well. And there have been, as you see here by the numbers of references cited in this systematic review, there have been many studies actually focusing at these on these prolonged neurological impacts of COVID-19. So the main debate nowadays, of course, or controversy, is whether these neurological complications derive from direct infection of the brain by the virus, or more likely, or at least supported by more studies thus far, due to a cytokine storm or the triggering of brain inflammation, perhaps as a response to peripheral inflammation induced by viral infection. So this is something that actually Fernanda de Felice has been continuing in collaboration with Fernanda Tovar-Moll from IDOR, a research institute in Rio. And they have been doing a follow-up, a very nice systematic follow-up of a number of patients that have experienced neurological manifestations in COVID-19, and they're trying to determine what kind of mechanisms may be behind those symptoms. However, we decided to take a slightly different approach. We decided to look at a possible bidirectional link. So rather than COVID-19 perhaps leading to neurological disorders, I went back to my previous interest in Alzheimer's disease, and I decided to ask whether perhaps Alzheimer's disease might predispose or be a risk factor for complications of COVID-19. And to address this question in a more straightforward way and with larger numbers of cases to study, we decided to do a study using a very powerful tool, which is the UK Biobank, which is this amazing public repository of information on about 500,000 UK participants. So we submitted a proposal to get data from the UK Biobank, and all the details of the study that we have performed thus far are described in this paper that was published a couple of, well, actually 4 or 5 months ago. And this was done by Ana Tahira, who is a postdoc in 𝗏𝗏𝗏𝗏𝗏's lab at the Butantan Institute in São Paulo. So this is published, but I just want to highlight 3 results from that study. The first one, what is the risk of having dementia, or in particular having Alzheimer's disease, in terms of infection by the SARS-CoV-2 virus? So here we look at various, a large number of potential risk factors in the UK Biobank cohort. And we find that, as has been well known, African ethnicity confers about a 2 to 3-fold increase in odds ratio. So down here, we find the odds ratio compared to white ethnicity. and so or to people not having these disorders listed down here. So African ethnicity confers some risk, but interestingly, that risk is higher for younger individuals, like between 66 and 74 years of age. But if we consider the oldest individuals in the cohort, those above 80 years old, one finds that actually African ethnicity confers a very small increase in risk to COVID-19. And all the other disorders that we looked at had very little effect or impact on the risk of COVID, of infection. However, when we looked at dementia, and in particular Alzheimer's disease, we found a very large increase, a six-fold increase in risk in Alzheimer's disease for infection by SARS-CoV-2. And then, of course, one of the reasons that that could be behind this is that people with Alzheimer's disease, especially those with more advanced forms of the disease, people need to have caretakers, or they live in long-term homes, long-term residences, and therefore that might increase the risk of infection. So just rather than just looking at infection as a readout, we decided to look at COVID-19 related hospitalization. So again, here we find that African ethnicity leads to an increase, significant increase, about threefold, in the risk of COVID-19 related hospitalization, but not of overall hospitalization. And then all-cause dementia, meaning other forms of dementia, and in particular Alzheimer's disease, they do not increase the risk of overall hospitalization due to other causes, but they cause a really dramatic increase in risk for hospitalization or aggravation of COVID-19 requiring hospital treatment. And finally, we looked at whether having dementia or having Alzheimer's disease would be an important risk factor for COVID-19 related death. And so again, we found in this case that dementia and Alzheimer's disease confer about a 2 to 3-fold increase in odds ratio for death caused by Alzheimer's disease. So the conclusion of this study is that having dementia, and in particular having Alzheimer's disease, not only increases the risk of infection, which might be explained by more trivial factors like exposure to the virus, as I mentioned, but they also trigger or increase the susceptibility of the individuals to COVID-related aggravation of the health, and especially to death. So meaning that these are important risk factors that should be considered. Now, what we're doing now, of course, is trying to determine why this takes place. Why is it that dementia or Alzheimer's disease in particular confer increased susceptibility to the aggravation of health by COVID-19? And to answer this question, we are now conducting a genome-wide association study, targeted version of that. Actually, we're looking at AD target risk factors, genetic risk factors for Alzheimer's, and trying to determine which of those, if any, might be related to COVID-19. And thus far, our initial analysis have really beginning to show interesting candidates, which we need to validate. So I'm not bringing those today to this presentation, but I will be happy to discuss later on if people are interested in that. So I just want to finish by thanking, I think I've thanked most of the key collaborators along the way during my talk, and the funding agencies that have supported my lab through the past few years. And thank you very much.
Okay, thank you very much, Professor Sergio. We have time for a couple of questions. So I have a question. So I thought it was really interesting, your last data about dementia and Alzheimer's disease as a risk factor for the severity of COVID-19 infection. So do you have any idea about the influences of the pharmacological treatment that's being given to patients, just like corticotherapy, just like dexamethasone, that could change the inflammatory process and maybe have an influence on these risk factors?
Yeah, I don't know if I don't think that would change the fact that AD or dementia are risk factors for COVID, but I definitely think that treatment with anti-inflammatory drugs, like as you mentioned, at the right time would be beneficial not only to the peripheral body, but also to the central nervous system. So I think the available data for infection of the brain by neurotropic viruses would support such a treatment avenue. Yeah.
Mm-hmm. So we have a question from Victoria that she's asking: is there an answer for this risk association between male sex and I think it's COVID-19?
Yeah. In fact, not that I know of. That it hasn't been really clearly established. Why? Just similar to the ethnicity that I mentioned. Why is it that African ethnicity or Asian ethnicity confers significant increases in risk factor compared to white ethnicity? But in those cases, as well as in the sex difference, there is clearly an increased risk with sex, has been shown in many studies, but not that I know of, that we don't know the answer to that. Why is it?
Mm-hmm. Yeah, so there are lots of open questions.
Yes, there are lots of open questions.
Mm-hmm. So if no one else I have one final question for you. So we know that this cytokine storm is a big concern and one of the main consequences of SARS-CoV-2 infection. And is there any advance or any data, or are your group interested in looking of if this cytokine production is happening in the brain, or if there is some kind of migration or spreading of this peripheral inflammation to the brain and then leading to these cases of encephalopathy that we are seeing in patients with severe SARS-CoV-2 infection?
So that's a very good question, Gilda, and one that, as I mentioned, the two Fernando de Felicici and Fernando Tovar-Moll, are conducting a follow-up study now of patients who were infected with SARS-CoV-2 and had COVID and had neurological impacts during disease. And so these patients are being followed, and one of the things they're looking at is trying to determine levels of these pro-inflammatory cytokines in the blood, as well as in the CSF, in the cerebrospinal fluid. And they are looking at possible correlations between these cytokines. So there is the results that they are obtaining suggest that indeed there is, of course, a peripheral cytokine storm, which is confirming those patients. And what they're trying to determine is whether these are now coupled to changes in cytokines in the CSF. That is, again, ongoing study, but I think the initial results from what I've seen from them is that, yes, there are certain cytokines that may be increased significantly in the central nervous system of these patients. So it could be that there is some sort of transport across the blood-brain barrier, for example, which is known to be permeabilized or defective under conditions of inflammation. And so that could lead to peripheral to brain movement of cytokines, and that would stimulate local production. But of course, it's still a bit early to know whether the brains of people that this died with COVID-19, for example, really had enhanced levels of cytokines. There are some studies going on, but it's still early on.
Mm-hmm. Okay.
So we still have more questions than answers, for sure.
Mm-hmm. Yeah, it's a really hot topic nowadays, so it's expected. So do you have time for a last question? I think so. So Sergio Schmidt is asking, although you have not focused on the DG and memory, I wonder if you are considering the parietal cortex and attention disorders, I think in the field of viral infections.
So yeah, so we focused on memory, but we also looked at, in the same paper on the Zika virus, for example, we focused on other brain areas, for example, areas that are involved in motor control, for example. We looked at actually at the entire brain. And there was, if you're referring to the Zika work that we did find distribution of virus all over the brain. It was more localized to the hippocampus and to the frontal cortex, as well as the striatum. So those were regions that we focused our attention more. It was very, very little present, for example, in the cerebellum. So there is some regional anatomical specificity to infection. It's not all over the brain, but it is possible that this would lead to other neurological impacts. We have not performed any tests of attention with these mice, other than the control tests that we do, showing that they, when we do the memory test, we do some control tests for exploration. So if the mouse was not exploring the objects in a proper manner, we would notice that would be apparent. And so but those were only really the very minor things that we did in terms of checking attention and interest, motivation, if you will. But we have not performed studies on attention tests.
Mm-hmm. Okay.
But I would not have a clear effect. Yeah.
Mm-hmm. So there are some more questions popping up right now, but I think we are short of time. We are a little bit late on the schedule. So I'd like to thank you, Professor, for your brilliant talk, and thank everyone for being here with us this morning. And I hope you enjoy the rest of our meeting. So thank you very much, and have a nice afternoon and have a nice weekend.
Thank you very much. Bye.